Hello, my name is Dr. Roderick L. Roll, and today we will start covering the portion of microbiology where we talk about how drugs affect microorganisms. Drugs are chemicals that can positively or negatively affect the physiology in any manner. Chemotherapeutic agents are drugs that act against disease. We will talk about the chemotherapeutic portion of drugs. Antibiotics are the ones that we will spend the most of this time discussing. These antibiotics are antimicrobial agents. They are a drug and they are produced by one organism, but they are toxic to another. For example, some bacteria, like streptomyces, will produce a chemical, and this chemical can retard the growth of another organism towards it. Therefore, it hinders the growth of other organisms. It is produced by one organism but affects another organism, and it affects it in a negative way. Therefore, some organisms are known to release chemicals. These chemicals are called antibiotics. In this picture we're looking at of yeast and a bacteria called Staphylococcus aureus. If you look, there is a clear halo around the yeast, and the scientific name of yeast is penicillium. So if you look at the penicillium that they're pointing to in the picture, you can see that halo. That halo means Staphylococcus will not grow in that zone. That zone is a zone of inhibition. The penicillium is releasing a chemical into the medium that is retarding the growth of Staphylococcus aureus. This picture is showing a similar process. So Selma Wakesman discovered that soil-dwelling bacteria in the genus Streptomyces released these chemicals, and they coined the chemicals the term antibiotics. In lab, you would do something just like this. You would create a bacterial lawn on a petri dish. We'll probably use maybe 500 microliters of pre-made broth that I contaminate it with bacteria. Once it is growing to a confluent level, you'll pull the 500 out and squirt it onto this petri dish and use a hockey stick to smear it. You're going to smear it to make it evenly distributed. Then you'll drop these pre-soaked discs that have been soaked in different antibiotics. And then you will measure those zone of inhibitions. Those pre-soaked disc has these chemicals on it that came from another bacteria that is going to negatively affect this bacteria. The gram-positive filamentous streptomyces category produces more than half of the known antibiotics. There are many different species of streptomyces. In the picture, I found an electron micrograph view of streptomyces growing. This list shows other microorganisms that produce antibiotics. The first portion looks at different fungi. You got penicillium again. The next section looks at bacteria. All of those different types of bacteria produce antibiotics. We're going to talk about several of these, like vancomycin, rapamycin, bacitracin, polymyxin, Gentamicin, we're going to talk about all those. Erythromycin, streptomycin, all of those we'll cover in the subsequent lectures. But what I want you to take from this is that most of the antibiotics come from a genus, a specific genre, which is Streptomyces. And we will also cover these antibiotics too, neomycin, um, tetracycline, and so on. Now we're going to get into the actual antibiotics. So this slideshow is titled Antibiotics that Affect Metabolism specifically. So the first drug discovered that could treat bacterial infection occurred in the 1930s, and it was an organic sulfur-containing molecule called sulfanilamide. I am pointing to this sulfur-containing compound, and you can see the S in the structure, and that S stands for the sulfur atom. Previously, we talked about a guy named Domack and his search for the magic bullet, and this was his magic bullet. Sulfonamides are structurally similar to p-amino benzoic acid. We're going to now call that PABA or PABA. So if you look at the top images, I have PABA, and then I have the chemical structure, molecular structures of the sulfur-containing units. These sulfur-containing units look so similar that they are called analogs. And the portion that is very similar, they've highlighted with like a orangish color. This PABA, the bacteria will naturally use it to produce DNA. PABA. How it helps the bacteria produce DNA is it first is going to be enzymatically converted into folic acid. So look directly under the images of PABA and their structural analogs and look at that flow chart. PABA is enzymatically converted into folic acid. That's the first step. Semisynthetic antibiotics have a slightly altered chemical structure from the original. So we can take these naturally produced compounds that come from bacteria or fungus, and we can modify it slightly to make it last longer and be more effective. So in the picture, outlined in red, you see penicillin to the left, where A is located. To the right of that, we have chemically altered it. Now that semi-synthetic penicillin is going to be longer lasting and more effective. As I mentioned, we're going to talk about how these antibiotics affect metabolism specifically. And I'm going to mention some of those sulfonamides, trimethoprim, and dapsone. So we'll talk about those three shortly. Next, after this slideshow, we'll cover some of these other ones that are circled. So let's expand on metabolism. So inhibition of metabolic pathways can inhibit microbial growth. So these are anti-metabolic chemicals. These are anti-metabolic antibiotics. They're going to somehow affect an enzyme that may be involved in growth, nutrition, the health of the microbe. The metabolic pathway involved in DNA synthesis of bacteria requires a molecule called PABA. I mentioned this before. Now I'll put some stars on this flow chart to illustrate how. So the first step is PABA is converted into dihydrofolic acid through that enzyme. The picture below is showing you the actual enzyme performing this task. It has an active site, and PABA has a shape that binds to the active site, and it will immediately be converted into dihydrofolic acid. Remember, these sulfa drugs are analogs to PABA. They look very, very similar. These sulfa drugs can compete for that active site, therefore inhibiting production of that product. So just look at image A labeled competitive inhibition. PABA is the red substrate that needs to be converted into a product, and the enzyme will perform that task. But you have this yellow inhibitor that is bound to the active site. As long as that inhibitor is bound to the active site, the substrate, which is PABA, cannot be converted into dihydrofolic acid. If dihydrofolic acid is not created, then the bacteria cannot make DNA. You have effectively inhibited metabolism or a metabolic pathway. This shows you every biochemical step to get to the synthesis of DNA. Notice in the picture below, this sulfonamide, is inhibiting the first step, whereas trimethoprim is inhibiting the step between dihydrofolic acid to tetrahydrofolic acid. Through competitive inhibition with the PABA analog, dihydrofolic acid production will be inhibited. This will ultimately stop the DNA synthesis. So this is a picture we've looked at several times already. So just follow the flow chart again. PABA needs to bind. It cannot bind because this analog is present. If the analog is present, you will not produce the preferred product, which is dihydrofolic acid. Will this affect us? No. So folic acid is vitamin B. So humans, we, while we're taking this drug that is going to be an analog for vitamin B, or be a analog for PABA, humans make their own, consume their own vitamin B, so it will not affect them, and they consume it in their diet, such when they eat leafy greens, asparagus, broccoli, and so on. So this will not, again, if you take those drugs, it will not affect the metabolism of our cells, only the metabolism of the bacteria. Newer sulfur drugs are going to be combination drugs. So instead of just having the sulfanilamide or the sulfur containing drug to the left, you're going to have two different drugs in combination. And I mentioned before how in that flow chart of about 4 or 5 individual steps, one of the drugs stopped the first step and the other drug stopped like the third step. So this first sulfur drug stops the synthesis of dihydrofolic acid, whereas, which is enzyme 1 in this flow chart, whereas the second drug, trimethoprim, it stops the synthesis of tetrahydrofolic acid, which is a second enzymatic reaction. So again, a combination therapy, if you take that basotrim, it is going to do a 1-2 punch to make sure you knock down the production of DNA and RNA in the bacterial infection. Other synthetic agents like sulfones are related to sulfonamides. They also interfere with folic acid production. These other examples, sulfones like dapsone, and I have a little star by the chemical structure of dapsone. It is an antibiotic. It is used to treat a mycobacterium. And remember, Microbacterium causes TB and leprosy.